back to Inspiring Minds. I am Rishabh, back again with another video. Guys, in the last video we discussed about the carbohydrate absorption. In this video we will discuss about the protein absorption. How the protein we eat from the diet will be metabolized and how it will be absorbed in the blood. Okay, so let's start. The protein we eat is the dietary protein. Okay, it is the dietary protein which we will eat. लेकिन ये protein कैसा है? This is a complex protein. So what we have to do first, we have to metabolize this complex protein, means we have to break it and then we will absorb it. Okay, so how it will be metabolized, how it will be break down, and then how it will be absorbed. So let's see. Now on this dietary protein, in this dietary protein we will have three enzymes. Okay, the action of three different types of enzymes will work and will break down this dietary protein into oligopeptides plus few amino acids. Okay, so the protein is what? Protein is the chain of the peptides, yani chain of the amino acids. More than 50 amino acids when combined forms the complex protein. This complex protein is a structure of oligopeptides. So when we will break this protein chain, we will get some oligopeptides. Oligopeptides is a, you can say, a chain of amino acids with 20 to 30 amino acids. Okay, and we will get also some free amino acids as well. Okay, so what will happen? First, let's talk about these three different types of enzymes. Okay, so types of enzymes. The first enzyme, which will be, it's the trypsinogen, which was released from where? The pancreas. Okay, this trypsinogen will be converted into the trypsin. All the enzymes about the GIT enzymes, I'm covering the absorption video of the first. I have discussed about carbohydrate. In this video we are talking about proteins. In the next we will talk about the fat absorption. And for these the basic concept you need is about the GIT enzymes, their targeted organs, they produce hormones and how they are converted. And you can see my earlier video about GIT enzymes on the same channel. Okay, you will get the link in the description. Now this trypsinogen will be converted into trypsin by enteropeptidases. These enteropeptidases you can get in the enterocytes in the intestine. Okay, now the first enzyme which we need is this trypsin. Okay, now what will happen? There are some inactive proteases where they are in the intestine only, actually in the duodenum, and they will be converted into its active form. And who will convert them into active form? The trypsin only. And this will be the second. And a very important example of this active form, it's which you know, it's chymotrypsin. Okay, the chymotrypsin is an example which you can see as a the trypsin has converted this inactive into the active form. Okay, now the third which we need, third are the brush bordered proteases. These is the number 3. So these three different types. The first is the trypsin, second is the active form of proteases like chymotrypsin, and third are the brush bordered proteases which are released from the villi like cells in the intestine. These three will act on what? These three will act on the dietary proteins, and this dietary protein will break down into the oligopeptide and amino acids. Okay, now what we need to discuss is there are two types of oligopeptides. One are the large peptides and other are the small peptides. Small peptides means 2 to 5 amino acid chain, and large means 10 to 15 amino acid chain, and amino acids means the single amino acids. Okay, how they will be transported? Now this whole process until we discussed where it was occurring in the enterolumen, means the lumen of the intestine, and all this has to go where? In the intestine cells, also called as enterocytes. So we have to transport these three into the enterocytes, and how they can be transported into the enterocyte is the main question. These large peptides will be transported through a process called transcytosis. Okay, this will be transported through transcytosis, a simple process in which the lumen will be transferred into the enterocytes. The content of the lumen will be transferred into enterocytes. These small peptides, how they will be transported? Through hydrogen ion co-transport. Means what? The hydrogen ion will be responsible for this transport. Means co-transport means what? There will be the coordinated, these small peptides along with the hydrogen ion will be coordinated and transported into the enterocytes. And the similar fashion will be performed for the amino acids, where they will be transported through sodium ion co-transport. Okay, sodium ion will be co-transported along with these free amino acids. Okay, so what we have to discuss is only three forms of I would say proteins will be transported. One is the amino acid, which will be transported through sodium ion. Small peptides will be transported through hydrogen ion, and large peptides will be directly transported through the transcytosis. And you should say there's a point where we have needed some co-transporter in the case of small things like small peptide and amino acid, but in the large form the protein is already being transported without anything help. So you can understand it by the way, like like proteins are very essential for us, and if the proteins are small in nature and they are being transported, they can be flooded out in the blood flow and can be wasted. They can be excreted out in the urine or in the form of urea, so they can be wasted. So we need some transporter to carry the small forms of protein, but the large peptides are so large that they can be transported directly. Okay, so they don't need any transporter. So now all these three things, where are they present? All these three things are present in the enterocyte. And now it's a simple case. Now they had been reached the intestine. After that they will reach the blood vessel and the absorption will be completed. So this way the absorption of the proteins occurs, in which we can revise very quickly. We eat the dietary protein. By the action of three different enzymes, these will be converted into oligopeptides and amino acids. What are these three enzymes? First is the trypsin. Second is the inactive to active form of proteases present in the intestine and the pancreas. And third is the brush bordered proteases, which are present in the duodenum. Then these oligopeptides are of two types, large and small, and also free amino acids. So we have ultimately three things which we will need, and how they are transported: transcytosis, hydrogen ion and the sodium ion. And then they are just absorbed in the blood vessel. They reach in the blood vessel and then ultimately reach to their target organ. So this was a short video again about the protein absorption. These are absorption of the items which we eat in a regular diet: the carbohydrate, the protein and the fats. And they are short concepts, very important in any you say clinical questions, and you should know everything about them. So that's it for this video, and I hope you understand everything. In the next video we will discuss about fat absorption. Okay, see you.